about 15 kilobase pairs. Because the DNA that corresponds to an average gene is about 1,000 nucleotides, each of the fragments of purified DNA has about 15 genes. A single cell usually incorporates only one or a few DNA fragments, so only a small proportion of the genes of the cell can be transferred to another by a single transformation event. Competence. A cell that is able to take up a molecule of DNA that can be transferred is said to be competent. Only certain strains are competent. The ability seems to genetically determine. Competence in most naturally transformable bacteria is regulated, and special proteins play a role in uptake and processing of DNA. These competences, specific proteins may include a membrane-associated DNA binding protein, a cell wall autolysin, and various nucleases. One pathway to natural competence in Bacillus subtilis are part of a quorum sensing system, a regulatory system that responds to cell number, regulated by the two-component regulatory system. Cells can produce and excrete a small peptide during growth and become competent in high concentrations of the peptide, though the action of COMP-P, a sensor kinase, and COM-A, a response regulator protein. COM-A is an activator protein that regulates a number of genes affecting transformation, and Bacillus, about 20% of the cells become competent and stay that way for several hours. However, in Streptococcus, 100% of the cells can become competent, but only for a few minutes during the cell growth cycle. Uptake of DNA. Bacteria differ in the form in which DNA is taken up, although in both cases only single-stranded DNA enters the cytoplasm. A Haemophilus, which is a gram-negative, for example, only double-stranded DNA is taken up into the cell, despite the fact that only single-stranded segments actually become incorporated into the genome by recombination. In gram-negative bacteria, it seems that double-stranded DNA is degraded to a single-stranded form in the periplasm space, and only a single strand is transported to a cytoplasm. In the gram-positive bacteria, Streptococcus and Bacillus, by contrast, only a single DNA strand is taken up, while the complementary strand is simultaneously degraded. However, in all cases, double-stranded DNA binds more effectively to the cells. During transformation, competent bacteria first bind DNA reversibility. Soon, however, the binding becomes irreversible. Competent cells bind much more DNA than do non-competent cells, as much as 1,000 times more, as we noted earlier. The sizes of the transforming fragments are much smaller than that of the whole genome, and this DNA is further degraded during the uptake processes. In Streptococcus pneumonia, each cell can bind only about 10 molecules of double-stranded DNA, of 15 to 20 kilobase pairs. However, as they are taken up, they are converted to single-stranded pieces of about 8 kilobases. The DNA fragments in the mixture compete with each other for uptake. And if excess DNA that does not contain a genetic marker is added, a decrease in the number of transformants occurs in preparations of transforming DNA. Only about 1 of 100 to 200 DNA fragments contain the marker being studied. Thus, in high concentrations of DNA, competition between DNA molecules results in saturation of the system. So even under the best conditions, it is impossible to transform all the cells in a population for a given genetic marker. The maximum frequency of transformation that has so far been obtained is about 20% of the population. Actually, the values usually obtained are about between 0.1 to 1.0%. The minimum concentration of DNA yielding detectable transformants is about 0.0001 micrograms, a millimeter, 1 times 10 to the power negative 5 micrograms, divided by milliliters, which is so low that it is undetectable chemically. Interesting in Haemophilus influence, there is a requirement that the DNA fragment have a particular base pair sequence for irreversibly binding uptake to occur. This sequence is found at an unexpectedly high frequent and homophilous chromosome. Evidence such as this, and the fact that at least certain bacteria become competent in their natural environment, suggests that transformation is not laboratory artificial but plays an important role in gene transfer in nature. Integration and transforming DNA. Transforming DNA is bound at the cell surface by a DNA binding protein, after which either the entire double-stranded fragment is taken up or the nucleus degrades. Our strand and the other is taken up. After uptake, the DNA associates with the competent specific protein that remains attached to the DNA, presumably preventing it from nucleus attack, until it reaches the chromosome where Rec-A protein takes over. The DNA is then integrated into the genome of the recipient by recombination process. During replication of the heteroduplex DNA, one parental and one recombinant DNA molecules are formed. One segregation at cell division, the latter is present in the transformed cell, which is now genetically altered as compound compared to the parental type. The preceding discussion pertains to only small pieces of linear DNA. Many naturally transformable bacteria are transformed only poorly by plasmid DNA because the plasmids must remain double-stranded and circular in order to replicate. Transfection. Bacteria can be transformed with DNA extracted from a bacteria virus rather than from another bacteria, a process known as transfection. If the DNA is from the lytic bacteriophage, transfection leads to normal viruses production and can be measured by the standard phage plaque. Transfection has become a useful tool in studying the mechanism of transformation and recombination because the small size of phage genomes allows for the isolation of nearly homologous population of DNA molecules. By contrast, in conventional transformation, the transforming DNA is generally a random assortment of chromosomal DNA of various lengths, and this tends to complicate experiments designed to study the mechanism of transformation. Artificially induced competence, high-efficiency natural transformation is found only in a few bacteria, Acintobacter and Azotobacter, Bacillus, Streptococcus, Haemophilus, Neisseria, and Thermus, for example, are easily transformed. Many prokaryotes are transformed only poorly or not at all under natural conditions. Determination of how to induce competence in such bacteria may involve considerable empirical study, with variation in culture medium temperature and other factors. However, to transfer DNA into cells for genetic engineering in this chapter, and also it was necessary to find a way to make Escherichia coli, a gram-negative organism, competent. It has been found that when E. coli is treated with high concentrations of calcium ions and then stored in the cold, it becomes transformable at low efficiency. Escherichia coli treated in this manner takes up a double-stranded DNA, and therefore transformation by plasmid DNA is relatively efficient. Why the calcium treatment works is not known, but this procedure also works with some other gram-negative bacteria. However, methods such as this for artificial induction of competence are being supplemented by a new method termed electroporation. DNA transfer by electroporation. Electroporation is a technique in which cells are exposed to pulsed electrical fields to open small pores in their membranes. When DNA molecules are present outside the cells during the electric pulse, they can then enter the cells through these pores. Electroporation requires sophisticated power supply because the pulses must be carefully controlled and last for only a millisecond. for only milliseconds. This technique has been used to transport DNA into a large number of different species of prokaryotes, both archaea and bacteria, and also into many eukaryote cells. Additionally, electroporation allows the experimenter to transfer plasmid directly from one cell to another if both are present during electroporation. Therefore, electroporation allows small molecules of DNA to come out of the cells as well as to go in. This type of transformation eliminates the steps required to isolate the plasmid from the first strain before introducing it into the second. Transduction. In transduction, DNA is transferred from cell to cell through the agency of viruses. Genetic transfer of host genes by viruses can occur in two ways. In the first, called generation transduction, host DNA derived from virtually any portion of the host genome becomes a part of the DNA of the mature virus particle and plane of virus genome. The second, called specialized transduction, occurs only in some temperate viruses. DNA form a specific region of the host chromosome is integrated directly into the virus genome, usually replicating some of the virus genomes. Transducing virus particles in both specialized and generalized transduction is usually defective as a virus because bacteria genes have replaced some necessary viral genes. Some generalized transduction. If the donor genes do not undergo homologous recombination with the recipient bacteria chromosome, they will be lost. They cannot replicate independently and are not part of the viral genome. In specialized transduction, homologous recombination may also occur. However, since the donor bacteria DNA is now actually a part of a temperate phage genome, there are two other possibilities. One, the DNA may be integrated into the host chromosome during lysogenization, and two, the DNA may be replicated in the recipient as part of the lytic infection. Transduction occurs in a variety of prokaryotes, including certain species of bacteria, Desulfovibro, Escherichia coli, and Pseudomonas rhodococcus. Not all phages can transduce, and not all bacteria are transducible, but the phenomenon is sufficiently widespread that it likely plays an important role in genetic transfer in nature. Generalized transduction. In generalized transduction, virtually any genetic marker can be transferred from donor to recipient. Generalized transduction was first discovered and extensively studied in the bacteria Salmonella enterica with phage, and has also been studied with phage P1 in Escherichia coli, an example of how transducing particles may be formed. Is given. When the population of sensitive bacteria is infected with the phage, the events of the phage lytic cycle may be initiated. During the lytic infection, the enzymes responsible for packaging viral DNA into the bacteriophage sometimes accidentally package host DNA. The resulting particle is called the transducing particle. On lysis of the cell, these particles are released along with normal virions, and so the lysis contains a mixture of normal virions and transducing particles. Because transducing particles cannot lead to a normal viral infection, they contain no viral DNA, they are said to be defective.